Alright, welcome back. In this video, we are going to talk about the specifics of what actually happens during the cell cycle. The previous video discussed some of the necessities of the cell cycle and some of the challenges that we have to kind of deal with as we go through it, but now we finally have all the information we need to talk about what is actually going to go on. So the cell cycle is just a orderly set of stages from the first division of a eukaryotic cell, so the original cell we call the mother cell, to the time the resulting daughter cells end up being divided out. So one mother cell is going to produce two identical daughter cells, is what we're going to see here. Now, of course, you can appreciate, as we said in the previous video, there has to be a lot of preparation here, including a lot of anabolic preparation. We need to synthesize a lot of new materials. We need to synthesize new proteins, new nucleotides, we need to synthesize new membrane, new organelles, and most importantly, we need to duplicate the chromosomes. So some things that we're going to see as the cell goes throughout the cell cycle, the cell will grow larger, and we owe a lot of this to new protein and new membrane synthesis. The number of organelles will roughly double. That's not something we have to get exactly right, but roughly we will see the number of organelles double. And then the DNA has to faithfully be replicated. Now this we do need to get exactly correct. So when I say faithfully replicated, I mean you need to make exact correct copies of the DNA that was there before. Now, to help you appreciate this, imagine that someone gives you a 1,000-page textbook and says, okay, you're going to take out a whole bunch of sheets of copy paper and you're going to write down exactly all of the text that is in that textbook. How confident are you that you would be able to do that without making a single mistake? You're probably not very confident about that, right? And that has nothing to do with you in particular. I couldn't do it. The person across the street couldn't do it. Nobody could do it. We would all make a bunch of mistakes, right? Now, what the cell has to do is it has to replicate the DNA without making mistakes. Now, that should give you an idea of what a massive undertaking this is. Because what we eventually want is we want our two daughter cells to be genetically identical to the original mother cell. That is only possible if the DNA is faithfully replicated. So in the cell cycle, we are going to see two major stages. Interphase is where the cell is going to spend most of its preparation time, and this includes several substages that we'll talk about. And then the mitotic stage, which includes mitosis, the divvying up of all the materials, and cytokinesis, the actual dividing of the two daughter cells apart from each other. So the cell cycle itself operates kind of like a timing mechanism on a washing machine. If you've only ever used some of the newer digital washing machines, you may not know exactly what I'm talking about. But some old washing machines actually had kind of a little dial that you would turn. You would turn it to the cycle that you want to start with, and then the dial will kind of slowly kind of turn back, kind of like an egg timer. And each stage of the wash cycle, the rinse, the spin, the drain, would last a predetermined amount of time before it moves on to the next thing. The cell cycle operates very much like this, with each of the different stages that we're going to encounter lasting what is supposed to be a predetermined amount of time, although that can change. So the cell cycle is also highly regulated, so the cell needs to make sure that each step happens with extremely high fidelity. In other words, we want to make sure that we're doing each preparation stage and each action without making any sort of mistakes or hang-ups. So failure to have sufficient oversight over the cell cycle, that leads to uncontrolled cell growth and division, and in a lot of cases, that is how we end up with cancer, and we'll say a lot more about that in a later video. So as we said, interphase is a big, long preparatory stage in which the cell is going to synthesize new raw materials and essentially is going to double in size. So cells will actually spend most of their time in interphase. If you consider a human cell, the entire cell cycle lasts about 24 hours, and 22 to 23 of those hours are actually spent in interphase, in preparation, and then only about an hour or so is dedicated to the actual division of the cell itself, the mitosis and cytokinesis. So, like I said, mitosis represents the divvying up of the materials that we duplicated during interphase into the two daughter cells. And then cytokinesis is an actual formal separation of the two daughter cells by pinching the plasma membrane between them so that we actually produce two separate cells, each with their own unique plasma membrane. So let's cover what we're going to accomplish in each of the three stages of interphase. These stages are called G1, S, and G2. G1 stands for gap 1, or the first gap phase. So here the cell is going to synthesize and accumulate nucleotides and proteins that are necessary for later on. If you recall back to Chapter 3 when we talked about biomolecules, nucleotides are the building blocks necessary for building nucleic acids like DNA and RNA. Obviously, we're going to need a lot of raw materials, a lot of nucleotides, since we're about to make copies of all of our chromosomes, so we gotta have raw materials for that. And then we're going to accumulate new proteins. We're going to make new amino acids, and then we're going to use those amino acids to make new proteins that are going to be necessary for the cell cycle as we go on. And then here the cell will also begin to synthesize new organelles by making new proteins and new membranes to stick together into making new organelles like mitochondria and lysosomes and things like that. S phase, or synthesis phase, is possibly the most important because this is where DNA replication is going to take place. Now, we will not actually talk about DNA replication until we get to chapter 14, I think it is. But all you need to know for now is that the number of chromosomes is going to effectively double. So we want to make exact, correct copies of all of our 46 chromosomes if we're talking about a human cell. So if we make copies of all the chromosomes, we should end up, instead of 46 chromosomes, but we should now have 92 chromosomes. The other major thing we're going to make here is that we are going to make a new pair of centrioles. The cell only had one pair of them before, but after S phase, the cell is now going to have two pairs of them. We'll say more about centrioles and centrosomes here in just a little bit. So a couple other things that we're going to make here. We're also going to make DNA-associated proteins like histones. So if we're going to make a whole bunch of new DNA, we have to have the spools that we need to wrap them up in. So as I said, chromosomes will enter S phase as one chromatid each, and then chromosomes will leave S phase with two identical sister chromatids that are stuck together at a central region called the centromere. I'll have a picture here in just a minute so I can actually show you how this is illustrated. And then in G2, the second gap phase, the cell will synthesize more proteins, including those involved in microtubule maintenance like alpha and beta tubulin. This actually has a lot to do with the centriole synthesis that we did in S phase. We're going to find out very soon that microtubules are actually a very critical component of mitosis and separating the chromosomes from each other. Now, when we get to mitosis, we have to think about the most important thing we can accomplish during mitosis is to equally distribute those chromatids of the pairs of sister chromatids that are stuck together currently. We need to equally distribute those chromatids to the two daughter cells so that each daughter cell inherits the correct number of chromosomes. So the thing to consider here, and I kind of mentioned this before, a human diploid cell has two copies of each of the 23 chromosomes, so each diploid cell has a total of 46. After S phase, these cells are now going to have a total of 92 chromatids, or 92 chromosomes. After mitosis occurs, each of the two daughter cells should then go back to having 46 chromosomes, two copies of each of the unique 23. So an important thing to consider also is that we need to make sure that each daughter cell has only two copies of each chromosome. It's not good enough for each daughter cell to just have 46 chromosomes. We need to make sure it's two copies of chromosome number 1, two copies of chromosome number 2, two copies of chromosome number 3, and so on and so forth. It's not enough to just have the right number of chromosomes. You have to have two of each one. Okay, so let's start walking ourselves through the entire cell cycle using a little bit of illustration magic. So the first thing, and there's going to be some repetition here because I put together this kind of illustrated set of slides separately from what I have together here in this whole video, so there's going to be some redundancy here, so forgive me for that. So the first thing we want to understand is that if a cell is going to replicate itself exactly, it needs to make duplicate copies of all sorts of things like organelles, like mitochondria, endoplasmic reticula, lysosomes, centrosomes, and so on, biomolecules like proteins and lipids and nucleotides and things like that. And then we're also going to duplicate the DNA. We're going to make duplicate copies of all of our chromosomes. So you can see all these things are currently color-coded. The chromatin is currently contained within the cell's nucleus. This cell is currently in interphase, so it has not reached mitosis yet. We've got some mitochondria, we've got some lysosomes, we've got ER, we've got Golgi, we've got all that good stuff. But for now, take note of the fact that the cell only has one centrosome, so that's a pair of centrioles. Interphase cells only have one centrosome, but we're going to see that that's about to change. So as I said, if these materials were not duplicated in interphase, then each daughter cell would only have half the materials the original parent cell started with. So that is a lot of what interphase is all about, making sure that our daughter cells have the same amount of stuff that the mother cell started with. So before we get into mitosis, let's talk a little bit more about what's happening in S phase. So we said that a human cell is diploid, so it has 2 copies of each kind of chromosome, so that's a total of 46. After S phase, this number is now 92. This is now a tetraploid cell. So after M phase is over, after mitosis is over, we're going to get back to a total of 46. The other thing that you can see here is that the chromatin, even after DNA replication, it is still contained within the nucleus, and we still cannot make out visible individual chromosomes. We are not going to be able to see chromosomes until we get to mitosis. So 2 logistical concerns that we're going to get to once we actually get to mitosis. We want to condense the chromosomes once we get there because chromatin, as it currently is arranged very much like kind of condensed spaghetti, it's going to be difficult to work with and separate apart from each other, and that's a big reason why we want to condense the DNA into chromosomes, which are much more compact and easy to move around. And then also you can see that the chromatin is currently confined in the nucleus. We cannot separate the chromosomes if all the chromosomes are stuck in such a tight space. So once we get to the early phases of mitosis, we're going to see the nucleus is actually going to break down and disappear. And this will actually give the chromosomes the freedom they need to move around to opposite ends of the cell. And they will be pulled to opposite ends of the cell by structures called mitotic spindles. Each centrosome that you see here is going to become a mitotic spindle, hence the importance of duplicating the centrosome during S phase. We want to have one centrosome at one end of the cell and the other centrosome at the other end of the cell, kind of like a north pole and a south pole that we are going to pull chromosomes towards either pole. So I kind of used this analogy on the previous slide, but imagine trying to split an entire plate of spaghetti exactly between 2 people. Even if you could eventually do that, it's going to be hard to do because you're basically moving individual noodles one at a time. Now imagine if you could actually compact all the noodles into chromosome-like structures. Well, then all you would have to do is move those big packages of spaghetti around until both people have an equal number. So the cell actually overcomes a very similar challenge by condensing the chromatin into much more manageable chromosome structures. These are much more easy to move around and much more easy to obtain a situation in which both daughter cells have an equal amount of DNA. So chromosomes duplicated during S phase are actually held together by a structure called the centromere. If you look at this chromosome here, this is actually 2 chromatids, what were 2 separate chromosomes held together. One of them is actually the original chromosome from either mom or dad, and then the other is the duplicated copy of it that was made during S phase. So this chromosome that you see right here, once it was duplicated, the copy is held together to the original copy by a structure called the centromere. The centromere you can kind of think of as kind of like glue that holds these 2 chromatids together. Now eventually we are going to separate these chromatids from each other. The original copy is going to go to one daughter cell and the duplicate copy is going to go to the other daughter cell. Hopefully that can help you appreciate why it was so very important that we make exact copies of the DNA. We don't want one daughter cell to have one kind of DNA and the other to have a different kind of DNA. And then the other thing that you're going to see here are protein complexes called kinetochores that assemble around the centromere on opposite ends of each of the sister chromatids. Now you can kind of think of the kinetochores being like handles, kind of like you have handles on a chest of drawers. It's kind of hard to open a drawer on a chest of drawers if there's nothing to grab onto. If there's no handle, it's going to be hard to open. Similarly, the chromosomes are not going to be moved around very easily if there is not some sort of handle that the cell can kind of latch onto and drag the chromosome around. So you can kind of think of the kinetochores being like handles that will make transporting the chromosomes around the cell a lot easier. And then finally, before we actually get into the nitty-gritty of mitosis, let's reconsider the centrosome. The centrosome is sometimes what we call the microtubule organizing center of the cell. It consists of a pair of centrioles made of a protein called gamma tubulin. And the most important thing here is that the centrosome is the structure from which all microtubules in the cell emanate. So that is why we sometimes call it the microtubule organizing center. So during S phase, we saw that the single centrosome was duplicated so that we end up with two centrosomes. This will allow us to separate our sister chromatids from each other because the centrosomes are going to migrate to opposite ends of the cell. They are going to reach out towards the chromosomes with their microtubules, and the microtubules are sort of like ropes that are going to attach to the kinetochore handles. And then as the microtubules get pulled back towards the centrosome, we are basically trying to lasso the chromosomes and pull them towards opposite ends of the cell. And that is going to be the major mechanism by which we separate the sister chromatids from each other to make sure that each daughter cell gets an equal number of chromosomes. Okay, so let's get into mitosis itself. So we are done with interphase, we're done with our DNA replication, we're done with our centrosome replication, we've duplicated all of our organelles, the cell is roughly double the size of what it was before. Mitosis is going to happen in 5 separate stages. The first is called prophase. So during prophase, this is the earliest stage in which you can actually see visible chromosome structures if you look under a microscope. Otherwise, you won't be able to see them because the DNA has not condensed yet. So during prophase, the chromatin condenses down into visible chromosomes, and the nuclear envelope is going to break down and disappear. You can see that we have our 2 centrosomes, and they are actually now going to migrate to opposite ends of the cell. Those opposite ends of the cells are what we are going to call the spindle poles, and that's going to be where our spindles are going to locate. Now, one thing I will kind of sidebar here on, it says in the fine print down here, if this were a human cell, there would be 92 separate chromatids to keep track of. Obviously, that makes the illustration a little bit complex and hard to keep track of. So for simplicity's sake, I've only drawn a total of 8 chromatids here. They appear as 4 butterfly structures, but keep in mind each butterfly shape is actually 2 copies of the same chromosome that are bound together by their centromere. These sister chromatids, the 2 halves of the butterfly structure, are going to be separated later on. Okay, so once we get to pro-metaphase, the nucleus is gone, and the centrosomes are now at opposite ends of the cell. So each of these is what we call a mitotic spindle, and they are at opposite ends of the cell, which is what we call the spindle poles. So kinetochore proteins have now assembled on each sister chromatid at the centromere. That's what those green structures are. And the microtubules in orange are reaching out from each centrosome to bind to each kinetochore to essentially capture the chromosomes. So the microtubules latch on to those kinetochore handle proteins, and now that the chromosomes have been captured, the microtubules are going to start getting pulled back towards the centrosomes, and essentially these chromosomes that you see here are getting pulled in opposite directions. So what this is going to lead to is, because those pulling forces are equal on either side, all the chromosomes are going to line up at the vertical equator of the cell. This equator is called the metaphase plate, and this is where we move on to the 3rd stage of mitosis, which is called metaphase. The 2nd stage, I forgot to say, was called pro-metaphase. So prophase, chromosomes are visible and the nucleus breaks down. Pro-metaphase, the centrosomes are at opposite ends of the cell, and we capture our chromosomes with microtubules. And then because of the pulling forces, once we get to metaphase, all the chromosomes should be lined up at the metaphase plate. Now, what you're seeing here is the reason why we are not yet separating our sister chromatids from each other is because the centromeres are still intact. Remember, the centromeres are kind of like the glue that holds the pairs of sister chromatids together. What we ultimately want is we want to pull the sister chromatids apart from each other, but we can't do that until we actually dissolve the centromere glue, which is actually something that we are about to do. So this is actually very important. Lining the chromatids up at the metaphase plate is actually very important because this is going to be necessary for the cell to pass one of the checkpoints of the cell cycle that we will talk about later, which is called the Spindle Checkpoint. So once we get our chromosomes lined up at the metaphase plate and everything is attached properly, we are ready to move on to the next stage. But first, let's consider what we just talked about. The sister chromatid pairs are being pulled from both sides, so why are they not being separated from each other? Well, like we said, there are several proteins that are involved in preventing this, and most of them have to do with the centromere glue that is holding them together. So we're not actually going to separate the sister chromatids until we clear the spindle checkpoint. So let's talk a little bit about how that's going to happen. First, let's talk about the nature of the glue that holds the sister chromatids together. They are held together by a ring-forming protein complex called cohesin. You don't need to remember the names of any of these proteins, just the whole thing is called a cohesion ring. These cohesion rings assemble around the sister chromatids that are held together and basically acts like little zip ties that holds them together. Now, if you think about a zip tie, if you receive a big shipment, and nobody likes dealing with zip ties because they're kind of hard to take off, and if you want to separate different things from each other in a shipment, you got to get those zip ties undone. So think about what you would do to break a zip tie. You might get out a knife to cut the zip tie, you might get out a pair of scissors. Either way, perfectly good suggestions, and that is almost exactly what the cell is going to do in order to separate our sister chromatids from each other and allow them to separate into the two daughter cells. So at the pro-metaphase to metaphase transition, an enzyme inside the cell called separase is going to become active. Separase is a protease enzyme. This is an enzyme that breaks proteins by breaking the peptide bonds that hold the amino acids together. And what this is going to do is once separase becomes available, it's going to act very much like a pair of scissors, and it's going to cut that cohesion ring and basically cut the zip ties off that are holding the two sister chromatids together. Now, it is important to understand that this only happens once we get all the chromosomes lined up at the metaphase plate. If we don't do that properly, then separase will not be able to do its job because it does not get activated. So with the cohesion ring now cut, there is no longer any glue holding the sister chromatids together. So those microtubules that you can see are pulling from both sides. Now we can actually separate the two sister chromatids, so this sister chromatid gets pulled towards one of the spindle poles, and this sister chromatid will get pulled towards the other. So this separation of sister chromatids is only possible because of the action of this enzyme called separase. Now, the timing of separase suddenly becoming active at the metaphase to anaphase transition, it's interesting, but I don't expect you to know all that much about it. So there's other proteins involved, but you do not have to worry too much about it. You're welcome to look it up, but I'm not going to be asking about anything other than what separase is doing. Okay, so once we dissolve the centromere that is holding the sister chromatids together, we can now see at anaphase, our fourth stage of mitosis, the pulling forces on the microtubules are now pulling the sister chromatids away from the metaphase plate. You can see this equator where the chromosomes originally were, and you can see evidence that they are getting pulled away from it. So as the microtubules retract and pull the sister chromatids towards each spindle pole, the chromatids get further and further away from the metaphase plate. And then once we get the chromatids separated towards the far ends of the spindle poles, we get to telophase, which is the last stage of mitosis. So the mitotic spindles are now going to disassemble. They're going to turn just back into regular old centrosomes. And the chromatids that we have pulled towards opposite ends of the cell are now going to be encapsulated in new nuclei. And then you can actually see evidence that we get what is called a cleavage furrow, which is the structure by which we are going to separate this big tetraploid cell into two diploid cells. So each daughter nucleus that you can see on either side now has a diploid count of chromosomes, which is good. So that should be 46 chromosomes if these are human cells. And then this big red ring that you see right here, this is a contractile ring made of actin filaments that is going to help us formally separate these two cells from each other by pinching off the plasma membrane that joins them together. So that leads us into cytokinesis, in which we formally separate the two cells, and now we have our two genetically identical daughter cells, identical not only to each other, but to the original mother cell, and they should now have an equal amount of cytoplasm and plasma membrane and pretty much everything else. And you can see each of these cells now has only one centrosome as well. So again, these cells are genetically identical to one another because they retain the copies of the DNA that were produced during S phase. So one cell is going to have the original copy, and then the other cell is going to have the duplicate copies. So that is my illustrated storytelling of how mitosis goes about. Here is a figure from your textbook. I don't like it quite as much, but it's the same information, and you can see everything is broken down by exactly what is occurring. So I don't think this should be too much new information here, so I'm going to go ahead and go past it. Now, something I want to talk about is the idea that the cell division, the mitosis process, is going to be largely the same in animal cells as it is in plant cells. So if you look at this comparison of mitosis happening in an animal cell on top and a plant cell on bottom, you can see that everything is very largely the same. You can actually see the condensation of chromosomes during early and middle stage prophase. You can see pro-metaphase, we're starting to try to get those chromosomes aligned at the metaphase plate. Then we finally do that at metaphase. We start to separate them out, and you can actually see the spindle poles forming at anaphase. And then you can see we're starting to get formal separation once we get to telophase. Now, once we get to telophase, this is going to be where we start to notice some differences between what's going on in animal cells versus plant cells. Animal cells achieve cytokinesis by forming a cleavage furrow, and we'll talk more about this here in a minute, but you'll notice the plant cell is not forming a cleavage furrow. It's forming something different called a cell plate. So we'll talk about this here in just a minute. First, let's consider cytokinesis as it happens in animal cells. Cytokinesis just refers to the division of cytoplasm and everything else. It is essentially a physical separation of the plasma membrane so that we produce our two daughter cells, and it allocates the mother cell's cytoplasm and resources equally to the two daughter cells. Each daughter cell will have its own plasma membrane to call its own. You can sometimes see evidence of cytokinesis starting late in anaphase, but more likely you're going to start to see it in telophase. And like I said, just as an appetizer, cytokinesis is going to be different in plant cells compared to animal cells. When animal cells do cytokinesis, they form a cleavage furrow, which is a big contractile ring of actin filaments, and it is going to separate the two cells from each other, the two daughter cells, by basically acting like pulling on a drawstring that is then going to kind of pinch off the plasma membrane and pinch it into two. So here you can see an illustration of animal cell cytokinesis in which the two cells here kind of in late telophase, you can see our cleavage furrow here, and as we get closer and closer to the end of cytokinesis, you can see the shared membrane between the two cells becomes thinner and thinner. This cleavage furrow, made of actin and myosin motor proteins, pinches off the plasma membrane until they're actually formally separated from each other. Okay, so let's finally talk about cytokinesis in plant cells. So think about what you know about plant cells. What do plant cells have that animal cells do not? That is going to complicate cytokinesis as we have talked about it so far. Now, if you recall, plant cells have chloroplasts, they have central vacuoles, but neither one of those really have anything to do with cytokinesis. So remember, cytokinesis has to do with the separation of plasma membrane. Now, what does a plant cell have just on the outside of its plasma membrane that prevents things like cell lysis? If you said the cell wall, you are correct. So the cell wall complicates the process of cytokinesis and ensures that plant cells cannot operate cytokinesis the same way that animal cells do. So cytokinesis in plant cells is going to begin with the formation of what is called a cell plate. So essentially what the cell plate is made of is many membrane-bound vesicles that are donated by the Golgi apparatus. And essentially what this is the construction of a new plasma membrane in between the two sides of the late telophase plant cell. If you've ever seen one of those old sitcoms like The Odd Couple, I'm probably really dating myself with that reference. If you've ever seen one of those sitcoms where two roommates get sick of each other and they decide they're going to like paint a line halfway down their apartment and they're going to say, okay, this side is my side, this side is your side. Well, the cell plate is basically like the line that is separating the cell in two. So basically we are constructing a cell membrane in between the two sides of the cell because we cannot form a cleavage furrow. So the membranes of the cell plate are then going to become the plasma membrane between the two daughter cells. Now these two plasma membranes on either side is going to have a space in between them that is called the middle lamella, and this space is eventually going to get filled with a newly constructed cell wall so that each daughter cell can not only have its own cell membrane but its own cell wall as well. And you can see an electron micrograph of exactly what's happening here as well. You can see the cell plate forming, and you can see that obviously this is necessary because of the cell wall that surrounds the cell membrane, something that plant cells have to deal with that animal cells do not. Okay, and then the last thing that I wanted to mention is the idea that the cell cycle is not something that all cells are going to be going through all the time. So cells will, as we're eventually going to see, they're going to have to pick and choose the correct spots to go through the cell cycle only when environmental conditions are favorable and only when specific signals are actually permitting cell division to occur. If cells did go through the cell cycle all the time, we would basically be big walking bags of cancer all the time. We said the cell cycle is very heavily regulated, and a lot of that regulation is cells being instructed when to go through the cell cycle and when not to go through the cell cycle. So cells that are not actively in the cell cycle are said to be quiescent or in G0 phase. This means they have exited the cell cycle and will only divide again when favorable signals are received and unfavorable signals are taken away. Now, some cells, like brain and heart cells, are actually permanently quiescent, meaning they will never ever divide again. So when you think about your brain cells and your heart cells, the fact that they are permanently out of the cell cycle, this can really make you appreciate the severity of things like heart attacks and strokes, since both of which involve the death of brain and heart cells. If someone has a heart attack, a big chunk of tissue in their heart is going to die, and that tissue will never be replaced by newly dividing cells because those cells are permanently quiescent. So someone who survives a heart attack or survives a stroke is going to have to live the rest of their life with a big chunk of their heart or a big piece of their brain missing. All right, that is going to finally wrap up our discussion on the cell cycle, interphase, and mitosis and cytokinesis itself. The rest of our videos are going to be discussing little regulatory tidbits and little consequences of when these things do not go appropriately. So join us when we get there, and I will see you next time. So long.